Okay, let's imagine for a second that you're a single cell. You're surrounded by a world that's, well, pretty much constantly trying to kill you. You've got nutrients floating by, which you need, but you've also got poisons and predators. So how do you let the good stuff in while keeping all the bad stuff out? It's one of the most basic challenges for life, and today we're going to look at the absolutely brilliant solution bacteria came up with billions of years ago.
Right, so it all really boils down to this one question. A cell has to eat, it has to pull in fuel to live, but at the exact same time, it's got to defend against this constant barrage of toxins, antibiotics, you name it. It's a life or death balancing act, and it's happening trillions of times a second all over the planet right now.
So what's the first line of defense in this constant battle? It's the bacterial outer membrane. The best way to think of it is like the wall of a fortress. It's this tough protective barrier that separates the cozy, controlled environment inside the cell from the harsh, totally unpredictable world outside.
And that's the fundamental problem for our fortress, isn't it? A solid wall keeps everything out, which is great for defense, but terrible for survival, because the cell would just starve. On the other hand, if you just throw the gates open, you're letting in poison right along with the food. So what you need is a smart, selective gateway.
And that solution, the hero of our story, is a tiny, absolutely ingenious piece of biological machinery called a porin. These proteins are basically the bouncers at the nightclub door of the cell, and let me tell you, they are incredibly good at their job.
So what exactly is a porin? Well, in the simplest terms, it's a protein that forms a channel, a little pore, right through that tough outer membrane. It acts like a molecular sieve, meticulously filtering everything that tries to get in. And its job description? Man, it's pretty impressive. First, it has to let in small nutrients. We're talking molecules under 600 Daltons. Don't worry about the number. It's just a unit for measuring how heavy a molecule is. But at the same time, it has to be a bodyguard, blocking bigger, harmful things like antibiotics. It also protects the cell from enzymes that would literally chew it up. And it even helps keep the cell from bursting if the water pressure outside changes too fast. It's a real multitasker.
Okay, so how on earth does one tiny protein pull all of this off? Well, the secret is in its structure. It's this incredibly unique and super efficient design, really a masterpiece of natural engineering. Now this is where things get really cool. Porins basically break the rules of normal protein design. See, most proteins that sit in a cell membrane are made of these twisty, coiled structures called alpha helices, but porins, they use something totally different, a beta barrel. The easiest way to picture it is to imagine a flat ribbon of protein that curls all the way around on itself to form this hollow, super stable tube. And here it is. This is that beta barrel we were just talking about. You can see how the protein chain folds back on itself again and again, in this case 16 times, to form the individual staves of the barrel. And right there, down the middle, that's the channel. That's where all the action happens.
But here's the thing, porins rarely work alone. They're social creatures. They typically team up in groups of 3, forming what scientists call a trimer. This setup makes them incredibly stable, locking them firmly into that fortress wall. It's like having a 3-lane superhighway for nutrients all bundled tightly together.
So let's quickly break down this amazing little machine. First up, you've got the outer loops. These are facing the outside world, doing the first security check on anything that comes by. Then on the inside, you've got these shorter, simpler connections. But here's the really crucial part, a special loop called L3 that actually folds down into the barrel. It creates this super narrow bottleneck, this eyelet, and that's the main checkpoint that decides what gets through. Finally, these things called aromatic girdles act like perfect little anchors, locking the whole thing into the membrane.
You know, for the longest time, the scientific assumption was that this barrel was just a completely rigid, unmoving structure, a simple, passive hole. But recent research has uncovered a hidden secret, a dynamic quality that is absolutely key to how it works.
So how'd they figure this out? Well, scientists looked at something called the B factor. When they create a 3D model of a protein, some parts of it can look a little blurry. The B factor is basically a measure of that blurriness. I love this analogy. Think of it as a wobble score. The higher the wobble score, the more that part of the protein is moving around. And this led them to ask a really fascinating question. Is this whole porin barrel a stiff, unmoving structure like everyone thought? Or are some parts of this tiny gate more flexible, more dynamic than others?
And the answer was a huge surprise, a total shocker. By looking at those wobble scores, researchers discovered that the porin barrel is really a tale of two walls.
The inner wall, the part that's facing the other porins and its little team of three, is incredibly rigid. But the outer wall, the part facing the rest of the membrane, it's much, much more flexible.
Okay, so this chart is super cool. It shows us the data behind that discovery. Each point is the wobble score for one of the barrel's 16 strands. Now look with me at the strands that form the inner wall. That's about beta strands 1 through 5, and then 15 and 16. See how their scores are low, really close to the center? That means they're rigid. Now, look at the outer wall, from strands 7 to 13. Boom! The scores jump way out, showing a ton more movement and flexibility.
So what the researchers are saying here is absolutely crucial. This isn't some random accident or a mistake in the imaging process. They concluded that this flexibility is a built-in, fundamental feature of the porin's design. In other words, it's flexible on the outside on purpose. Okay, so one side of the gate wobbles more than the other. Big deal, right? Who cares? Well, it turns out that understanding this one tiny detail completely flips our old idea of a simple, rigid hole on its head. And it has massive implications for everything from basic biology to the future of medicine.
Just think about it for a second. What's the advantage of having a flexible outer wall? Why would that be a good thing for the bacterium? Well, it suggests the gate isn't just the static hole. It's a dynamic structure that can actually interact with its environment, maybe changing its shape just a little bit, to let certain things through, or maybe to wedge itself into the membrane more effectively.
And this is why it really matters to us. Knowing how these gates work, and more importantly how they flex, lets scientists get clever. We can design smarter antibiotics that are built to sneak through these flexible gates. Or we could design molecules that jam them shut and starve the bacteria. We can even take these super stable proteins, like another porin called ampg, and re-engineer them to act as tiny biological sensors. And since those outer loops are just hanging out there on the surface, they make perfect targets for developing new vaccines.
The porin is just a masterclass in biological engineering. It's a simple barrel that solves an incredibly complex problem with elegance and this hidden dynamic efficiency. And as we keep unlocking its secrets, it leaves us with this really tantalizing question. If we can truly master the design of these tiny biological gates, what incredible new doors in medicine and technology could we unlock next?